Hello friends, this is Dr. Pankaj Kumar, your mentor, and in today's sessions we will be talking about the very first chapter of biological classification, and that is bacteria. Right? The bacteria is a part of Monera. You know very well that what is Monera? Monera are a group of organization having the prokaryotic organization, right? It can be unicellular, it can be multicellular. So be very clear that the concept of unicellularity and multicellularity is not. The only thing what we need to keep in our mind that Monera is a prokaryotic organization. Okay. Now again in Monera you see, depending upon the nature of the cell wall, the two types of monerans has been recognized. What we call eubacteria and second what we call archaebacteria. Now what are the criteria? As I told you, the nature of the cell wall, right? So what is true bacteria? The eubacteria is a true bacteria. Now what are the character for the true bacteria? The character for the true bacteria is that in case of true bacteria, their cell wall is made up of the peptidoglycan. But in case of archaebacteria, their cell wall is not made up of peptidoglycan. That is a combination of some other type of the polysaccharides. Right? So that is how we differentiate between the archaebacteria and the eubacteria. In today's sessions, we will be talking about eubacteria or a typical bacteria. So first of all, let's talk about some of the very common features, right? So some bacteria are photosynthetic, right? And they possesses bacteriochlorophyll. Mind it, this bacteriochlorophyll is entirely different from the chlorophyll A. Chlorophyll A is a universal pigment. All photosynthetic plants have a chlorophyll A, but not the bacteria. In case of bacteria, in place of chlorophyll A, there is a bacteriochlorophyll. Again, you see, there are two group of, or what we can say, there are two categories of the photosynthetic bacteria, what we call purple sulfur bacteria and the green sulfur bacteria. Now, in case of purple sulfur bacteria, what we see, there is a presence of bacteriochlorophyll, right? But in case of the green sulfur bacteria, it is not bacteriochlorophyll. Rather, in place of bacteriochlorophyll, there is a chlorobium chlorophyll, which is also known as the bacterioviridin, right? So this bacterioviridin is actually present in a green sulfur bacteria, right? Now let's talk about their shape. First of all, the bacterial shape in general varies from 0.2 to 1.5 millimeter in diameter and 2 to 10 millimeter in length, right? Primarily, that occurs in three shapes: the coccus, the rod shape, what we call bacillus, in the spiral, or the angular shape, or what we can say the vibrio or the comma shape, what we can say, right? Have a look at this diagram, right? You can see in the diagram. Right now you can see this is a bacillus, this is a coccus, right? And this is a spiral. Okay. Now, in addition to that, there are some other information. Well, let me add over here. This information is not very important from the NEET perspective, just for the general knowledge that what is the smallest bacteria, what is the largest bacteria, and what is the longest bacteria. So these names are in front of you. Okay. Let's talk about the bacterial structure. In case of bacterial structure, this entire structure can be discussed under three broad heads. The very first is surface appendage, where we discuss about flagella and the fimbriae, right? The second, what we call the cell envelope, that include glycocalyx, cell wall, and the plasma membrane. And finally, the protoplasm. In case of protoplasm, what we see that what is the genetic material, what is the cytoplasm that is present in a cell. So these are the things what we study. Have a look at this diagram. So that is a typical diagram of a bacterial cell. You can see there is a genetic material, mesosome. Mesosome is nothing but an extension of the plasma membrane, right? So one by one, we will discuss about that, right? So first of all, let's talk about the flagella. Bacterial flagella consists of primarily three parts. That is filament, hook, and basal body. Basal body is attached with the plasma membrane. Now, chemically, it is made up of a protein called flagellin, right? And the basal body, as I told you, it is connected to the plasma membrane. Now, basically, it shows 9+0 structure. Now, what do you mean by 9+0 structure? There is a 9 duplet structure that is present at the periphery, and in the central region, there is no structure. That's why you call 9+0 structure. Now, why it is significant? Because if you talk about the eukaryotic flagella, in case of eukaryotic flagella, we have a 9+2 structure. But in case of the bacterial flagella, we have a 9+0 structure, right? And unlike our hair, it grows at a peak. That is the important thing. So we have to keep in mind that flagella always grows at peak. It is also a hollow structure, right? So you can correlate these two things, that it always grows at peak and it is a hollow structure. That is about the flagella. Okay. Now, depending upon the position of flagella among the bacterial surface, the different types has been recognized. For instance, it could be atrichous when there is no flagella. It could be amphitrichous when one flagellum at both ends. It could be monotrichous if only one flagella is attached at only one end, right? It could be cephalotrichous when two or more flagella attached to the end, right? It could be lophotrichous where two or more flagella attached at both ends. It could be peritrichous when flagella is attached at the entire surface, right? So these are the types of flagella entirely depending upon the position of flagella on a bacteria. Have a look at this diagram. So this diagram will give you a very clear idea that what is peritrichous, what is atrichous, what is amphitrichous, and what is lophotrichous, right? In NEET, sometimes the questions are asked on the very different types of flagella based on their respective positions. Okay. Now let's talk about one more thing, that is pilus and fimbriae. Mind it, the pilus are elongated tubular structure that is made up of a protein what we call pilene. Flagella was flagellin, here it is a pilene. Now, pilus is reported only on a gram-negative bacteria, right? And what is their function? It helps in the formation of conjugation tube, right? During the genetic recombination, and one of the type of genetic recombination is a conjugation. Okay. On contrary, if you see, the fimbriae are a small bristle-like fiber, right? It helps bacteria in attaching to the solid surface of the host tissue, right? Have a look at this diagram. So this will give you a very clear idea. So this outer structure you can see there, this is a pilus, right? And this fimbriae is a simple structure that helps in the very attachment process. Okay, so that is about pilus and fimbriae. Let's talk about the glycocalyx. Now, glycocalyx is the outer covering. Now, first of all, glycocalyx may or may not be present in bacteria. Chemically, it is made up of polysaccharides, right? Again, you see, there are two types, or we can divide glycocalyx in two types. One, the capsules, and second, the slime layer. Okay. Now, slime layer is nothing but it is a gel-like substances, and capsule somewhat a rigid substances. Again, capsule can be divided into two parts, what we call microcapsule and macrocapsule, entirely depending upon their width. Okay. So you see, slime layer basically helps in protecting the cell from the loss of water, right? At the time of desiccation, the water content from the slime, actually the bacteria takes them from the slime. Okay. But at the same time, it also helps attaching the bacteria to the host. That is equally important, right? Now, the capsule is responsible for giving gummy and sticky nature to the cell. So this is the feature for the glycocalyx. So the key point, it may or may not present, number one. Number two, chemically it is made up of polysaccharides. So these are two things what you need to keep in your mind. Now have a look at this diagram. So this diagram will give you a very clear idea. The outside there is a slime layer, right? So it is a gel-like substances, and as I told you, it helps in just taking the water or attaching the bacteria to the host. So these are the two main functions that it performs. Okay. Now let's come to a very interesting questions. I'm sure all of you know about mouthwash, and mouthwash are primarily used by those persons who have a very bad smell. And one of the reasons for the bad smell is one disease called pyorrhea, right? So pyorrhea is nothing but it is a bacterial disease. And what happens? That bacteria attaches to your gum, right? And when it attaches to the gum, this bacteria is going to give you a foul smell, and that foul smell is coming along with your breathing. Okay. Now how this mouthwash works? Mouthwash is nothing but it is a hydrolytic enzyme. Now what it does? It hydrolyzes the capsule or the slime layer. So you know very well, the bacteria is going to attach our gum with the help of slime layer, right? So if the mouthwash is going to remove the slime layer, so naturally bacteria cannot be attached to any surface, right? So thereby, the moment we use a mouthwash, immediately the bad smell goes. And now I'm sure all of you understand the reason behind that. Okay, so you can see there that this is the area, that is the mouth or the teeth suffering from a disease called the pyorrhea, right? And from there is a large number of bacterial growth, and that actually causes a foul smell, right? So that's all as far as the introduction of the bacteria is concerned. In the next, we will be talking about the cell wall, perhaps the most important part of the entire bacteria. Do join me in the another session of NEET Bites, where we will be talking about the bacteria. Thank you.